Welcome, curious minds, to the deep dive. Today, we're embarking on a truly magnificent journey. Not across continents or through time, exactly, but into a world far more ancient, infinitely more complex. The hidden universe within us all.
It's everywhere, isn't it?
Exactly. Imagine, if you will, a microscopic realm, just bustling with activity, yet utterly invisible to the naked eye.
Mm.
A world so fundamental it underpins all life as we know it. Every breath you take, every thought you have. It all comes back to this.
We are, of course, talking about the cell.
The cell, yes.
It's quite astonishing when you pause to consider it, really. Our mission today, then, is to sort of peel back the layers of this microscopic universe.
Right.
We're not just describing what's there, though. We want to tell the incredible story of how we came to understand it.
Yes, the story. From its very first humble glimpse to unraveling its intricate machinery.
It's a real saga of human curiosity, isn't it?
That's right. Think of it like unearthing chapters from some epic scientific mystery, a story woven with, groundbreaking observations, relentless pursuit of knowledge.
And some truly surprising aha moments along the way.
Definitely. We'll be uncovering the answers to some of life's most profound questions, one tiny structure at a time. And I promise you, the journey itself is as captivating as the destination.
Indeed. What makes this story so compelling, I think, is that the cell wasn't just discovered in one go.
No.
Its secrets were revealed really slowly, over centuries, piece by painstaking piece. Each observation building on the last, like solving a colossal puzzle.
Okay, let's begin this fascinating journey right at the dawn of discovery. Our story opens in, 1665?
1665, yes, with a brilliant English scientist, Robert Hooke.
Picture him leaning over his new microscope for the time.
State of the art back then.
Right. Peering intently at something as simple as a thin slice of cork. And what did he see?
These tiny, orderly, box-like compartments.
He named them cells, drawing from the Latin word cella, meaning little room.
It's quite a poetic start, isn't it, for scientific nomenclature? Like finding tiny rooms in a piece of wood.
Like a honeycomb, almost, full of miniature chambers.
Exactly. But just a few years later, 1674, a Dutch draper named Anton van Leeuwenhoek picks up the baton.
And his microscopes were something else entirely, weren't they?
Oh, vastly improved. Homemade, but powerful. And he experienced a thrill that must have been just unparalleled.
Finding life.
Yes. He was the first to discover what he called animalcules, these free-living cells in pond water.
Can you imagine the sheer astonishment? Seeing life move independently, swimming and dancing in a drop of water that looks perfectly still.
It must have radically broadened the understanding of where life could even exist. Suddenly, it was everywhere.
Okay, so we have these rooms, then things start getting more complex inside, right?
Precisely. Fast forward quite a bit to 1831. Robert Brown makes another key discovery.
The nucleus.
The nucleus, yes. He found this darkly colored, spherical or oval dot-like structure, usually near the center of the cell.
So the cell wasn't just an empty room anymore.
No, it was starting to reveal its internal command center, you could say. And building on that, in 1839, Purkinje coined the term protoplasm.
Protoplasm, for the stuff inside.
Exactly, for the jelly-like fluid substance that filled the cell. This was a crucial conceptual leap, recognizing that the cell wasn't just a box with a dot.
But a dynamic living material.
A bustling internal environment, yes.
Wow. Okay, so all these individual discoveries, they must have started coming together at some point.
They did, leading to a really profound realization, a unifying moment in biology. The cell theory.
Ah, Schleiden and Schwann.
That's them, two German scientists. They made the groundbreaking statement, all plants and animals are composed of cells, and that the cell is the basic unit of life.
That's huge. It wasn't just a discovery. It was like biology's theory of everything, right?
Yeah.
Uniting the whole biological world.
Absolutely. And then, sort of completing this foundational understanding, Rudolf Virchow added his vital expansion.
Which was?
Omnis cellula e cellula. All cells arise from pre-existing cells.
Meaning life doesn't just pop out of nowhere.
Exactly. It comes from what was already there.
Yeah.
Establishing this continuous, unbroken lineage of life stretching back, to its origins.
Incredible. But seeing inside these cells in detail, that needed another leap, didn't it?
Oh, a massive leap. The real game changer, the visionary leap that truly let us see the inner workings came much later. 1940.
The electron microscope.
The electron microscope. It wasn't just an improvement, it was a revolution. It finally made it possible to observe and understand the incredibly complex structure of the cell, its various organelles.
It's like before, you could see a city from miles away, maybe just the rooftops.
And suddenly you could walk its streets, enter its buildings, see the infrastructure, the traffic, everything. It opened up a whole new dimension.
So, after that incredible journey of discovery, what makes a cell a cell? What defines it?
Well, connecting it all, it boils down to its fundamental nature. It is the basic, irreducible unit of life.
Right. And one of the most elegant things, I think, is how their shape and size aren't just random.
Not at all. They're primarily dictated by their specific function. Form follows function beautifully.
Like a long, skinny nerve cell for sending signals.
Versus a round, fat cell for storage. Completely different shapes for completely different jobs. No two jobs are exactly alike, so the cells doing them are uniquely designed.
Which brings up another point. Not all life is made of many cells, is it?
No, definitely not. You have single-celled or unicellular organisms, things like amoeba, chlamydomonas, paramecium.
Masters of self-sufficiency.
Absolutely, performing all life functions within that one tiny unit. Sensing, feeding, moving, reproducing, it's all in there.
And then you have us, multicellular organisms, where cells group together, specialize.
Assume different functions, form tissues, organs, complex body parts.
But here's a truly mind-bending fact, isn't it?
Yeah.
Every single multicellular organism, no matter how huge or complex,
Started from just one cell.
from one come many. It's just incredible, the power of division and differentiation.
It really emphasizes the foundational role of that initial cell. And think about something simple, like looking at onion peels.
Okay.
If you take peels from onions of different sizes, a small one and a large one.
Their cells look the same size, right?
Exactly. Remarkably consistent. A larger onion doesn't have larger cells, it just has more of them. A fascinating consistency.
So wrapping this part up, we can confidently say, the cell is the fundamental structural unit of living organisms.
It's the basic building block, the essential component. All life is constructed from them.
Okay. So if we were to peek inside almost any cell, what are the absolute must-have features we'd find?
Under a microscope, you'll generally find three main indispensable features: the plasma membrane, the nucleus, and the cytoplasm.
Let's unpack those, starting with the outer boundary. The cell's border control, the plasma membrane.
It's much more than just a bag, though. It's an incredibly sophisticated gatekeeper, a dynamic barrier.
What makes it so special?
It's selectively permeable. That's the key. It isn't just passive. It actively decides what gets in and what gets out.
Permits some things, prevents others.
Precisely. It's a constant negotiation, vital for maintaining the cell's internal balance. Its survival depends on it.
And it manages this movement in clever ways, like diffusion.
Diffusion is a big one. It's spontaneous movement from high concentration to low. Think about how a scent spreads in a room.
Or how oxygen gets into our cells.
Exactly. Vital gases like oxygen and carbon dioxide move across the membrane largely by diffusion, effortless, down the concentration gradient.
Then there's osmosis. That's related, but specific to water.
Yes, osmosis is critically important. It's specifically the movement of water molecules through that selectively permeable membrane.
Which leads to interesting effects depending on the surrounding solution. The water dance, as you called it earlier.
Yes. If the surroundings have a higher water concentration than the cell, a hypotonic solution, water rushes in, the cell will gain water and swell up.
Like an overfilled balloon.
Potentially, yes. Conversely, put a cell, or even something like a deshelled egg, in a concentrated salt solution, a hypertonic solution.
It shrinks.
It shrinks dramatically.
Oh.
Water passes out of the egg into the salt solution. It beautifully shows the membrane's role in managing water balance, preventing bursting or shriveling.
And some cells use the membrane in really active ways, like the amoeba eating.
Oh, endocytosis. It's fascinating. Enabled entirely by the membrane's flexibility. It literally engulfs its food particle.
Wraps around it.
Wraps its membrane around it, like an arm reaching out, and brings it inside. Amazing flexibility.
And what gives it this flexibility? What's it made of?
Primarily lipids, fats, and proteins, arranged in this fluid dynamic double layer, the fluid mosaic model.
Okay. Now, plants have something extra on the outside, don't they? The cell wall.
They do. Unique to plant cells and also fungi and bacteria. This rigid layer sits outside the plasma membrane.
That's tough.
Incredibly tough. Its main component is cellulose. It provides amazing structural strength, allowing plants to stand tall.
And it helps with that water pressure problem too, right? Prevents bursting.
Exactly. A crucial defense. The cell wall lets plant cells withstand very dilute external media, gain lots of water via osmosis, but not burst because the wall pushes back.
So what happens if a plant cell loses water?
That's when you see plasmolysis. The living contents, the plasma membrane, and everything inside shrink away from that rigid cell wall. It's a very visible effect of water loss.
Okay, moving inwards again, the command center we mentioned earlier, the nucleus.
Back to the nucleus, that darkly colored, usually spherical structure enclosed by its own double-layered membrane.
And inside, the genetic blueprints.
That's where they are, in the form of chromosomes, though you only really see them as distinct rod shapes when the cell is about to divide.
And these chromosomes contain DNA.
DNA, yes, deoxyribonucleic acid. It holds all the information for inheritance meticulously coded, the secrets to life.
And specific parts of the DNA are genes.
Precisely. Genes are the functional segments of DNA. They contain the specific instructions for constructing and organizing the cell, telling it how to grow, what proteins to make, how to function, like individual recipes in the cell's huge cookbook.
What does the DNA look like when the cell isn't dividing?
It's less organized then. It exists as chromatin material, an entangled mass of thread-like structures, like a ball of yarn, basically, waiting to be neatly wound into chromosomes when needed.
So this whole setup nucleus, membrane-bound bits, is that universal?
No. This brings us to a really fundamental divide in the living world. Prokaryotes versus eukaryotes.
Okay, what's the difference?
Prokaryotes are simpler. Their cells lack a proper nuclear membrane. Their genetic material, usually just a single chromosome, floats in a region called the nucleoid, but it's not enclosed.
And no other internal compartments?
Generally, no membrane-bound organelles either. Think of them as efficient, open-plan workshops. Bacteria are a key example.
And eukaryotes, like our cells.
Like our cells, yes. Eukaryotic cells have that distinct nuclear membrane protecting the DNA. And they have a whole suite of membrane-enclosed organelles, each doing a specific job. They're generally larger, more complex, like a compartmentalized city.
Right, the city. Let's explore that inner city then.
Yeah.
The cytoplasm and its organelles.
The cytoplasm is everything inside the plasma membrane, but outside the nucleus. It's the fluid jelly, the cytosol, plus all the organelles suspended within it.
And it's far from just jelly, right? It's bustling.
Absolutely teeming with activity. And these specialized workers, the cell organelles, carry out all the different functions needed for life. Each has a critical job.
Okay, let's tour some key departments. How about the endoplasmic reticulum, the ER?
Good place to start. It's like the cell's factory and internal highway system, a network of membranes. Comes in two flavors.
Rough and smooth.
Exactly. Rough ER, or RER, is studded with little dots called ribosomes.
And ribosomes make proteins.
They do. So the RER is a major site of protein manufacture, especially proteins destined for membranes or export.
And the smooth ER. No ribosomes.
Correct. The SER is involved in different things like synthesizing lipids, fats, and steroids, and also detoxification in some cells. And get this, the ER even helps build the cell's own outer membrane, the plasma membrane.
How?
Through a process called membrane biogenesis, it manufactures some of the necessary proteins and lipids and incorporates them. It's building its own house.
Clever. Okay, next stop, the Golgi apparatus.
The Golgi. Think of it as the cell's packaging and dispatch center, or maybe the post office and finishing school combined.
What does it do?
It receives proteins and lipids from the ER, modifies them, sorts them, and then packages them into little vesicles for delivery.
Delivery where?
To various targets. Some stay inside the cell, some go to the plasma membrane, some are shipped right out of the cell. The Golgi also makes lysosomes.
Ah, lysosomes, the suicide bags. Why that dramatic name again?
Because they contain powerful digestive enzymes. They're the cell's recycling center, breaking down waste materials and cellular debris. If the cell gets seriously damaged or old, the lysosomes can rupture and release those enzymes, digesting the cell from within. A controlled self-destruction.
Grim but necessary, I suppose. What about energy? The powerhouse.
Mitochondria, the undisputed powerhouses of the cell.
Why a powerhouse?
Because they perform cellular respiration. They take fuel molecules like glucose and oxygen and release the energy stored within them.
In the form of ATP.
Exactly. ATP, adenosine triphosphate, that's the energy currency the cell uses for pretty much everything. Muscle contraction, nerve signals, building molecules. It all runs on ATP made primarily by mitochondria.
And mitochondria are special, aren't they? Like they have their own...
Their own DNA and ribosomes. Yes, it's fascinating. They're one of only 2 organelles in eukaryotes that do. It suggests an ancient origin, perhaps as free-living bacteria that were incorporated into larger cells billions of years ago.
Wow. And what's the other organelle with its own DNA?
Plastids, found only in plant cells and some algae.
And they do.
Different things depending on the type. The most famous are chloroplasts.
Photosynthesis.
Right. Chloroplasts contain chlorophyll, the green pigment that captures sunlight energy to make sugars. They are absolutely vital, the foundation of most food chains.
So plastids, like mitochondria, have their own DNA too.
They do. Chloroplasts also have their own DNA and ribosomes, again pointing to that possible ancient symbiotic origin.
Any other types of plastids?
Yes, there are also chromoplasts, which contain other pigments giving fruits and flowers their colors, and leucoplasts, which are typically for storage starch, oils, proteins.
Okay, one more major component, vacuoles.
Vacuoles, essentially membrane-bound sacs functioning as storage tanks. Animal cells might have small ones, but plant cells,
They have that huge central one, right?
often enormous. The central vacuole in a mature plant cell can occupy 50 to 90% of the cell volume.
Just for storage.
Storage of water, nutrients, waste products,
Yeah.
but also crucial for maintaining turgor pressure. It pushes against the cell wall, providing rigidity and support to the plant, helps keep it from wilting.
So many intricate parts working together. Now, how does one cell become many? How do they multiply?
Through cell division, there are 2 main types.
Mitosis.
Mitosis is the process most cells use for growth and repair. If you get a cut, new skin cells are made by mitosis.
And the result?
One parent cell divides into 2 genetically identical daughter cells. They have the exact same number of chromosomes as the parent, a perfect copy.
Okay. And the other type, meiosis.
Meiosis is different. It's specifically for producing gametes, sperm, and egg cells for sexual reproduction.
How is it different?
It involves 2 rounds of division, and the end result is 4 new cells. But crucially, each has only half the number of chromosomes as the original mother cell.
Half the number. Why?
So that when a sperm and egg fuse during fertilization, the resulting offspring gets the correct, full number of chromosomes, half from each parent. It also shuffles the genetic deck, increasing diversity.
Essential for reproduction and evolution, then.
Oh.
So after looking at all this, if we had to pick one primary difference between a typical plant cell and an animal cell,
The most obvious, structurally, is the cell wall, present in plants, providing that rigid support.
but absent in animal cells.
Typically absent, yes. That single difference has huge consequences for how plant and animal tissues are structured and function.
Wow. We've really covered a lot. From Hooke's simple quark slice
Through Leeuwenhoek's
to the
animalcule,
discovery of the nucleus, the cell theory,
the electron microscope revealing organelles like mitochondria and chloroplasts.
and finally, how cells divide through mitosis and meiosis.
It really hammers home that the cell truly is the basic building unit of all life, a whole universe compressed into this tiny package.
The fundamental blueprint.
Indeed. And understanding these tiny units, it profoundly impacts how we understand everything else in biology: disease, genetics, evolution.
The unseen world really does shape everything we see and experience.
It does.
What's truly captivating here, I think, is that even with everything we've uncovered, the cell still holds secrets. We're constantly learning more.
Always more complexity, more elegance being revealed.
So here's a thought to leave you with. If a single cell can perform so many complex functions with such intricate machinery and precision, what does that tell us about the hidden potential in the smallest parts of our world? And perhaps even within ourselves.
A profound question. It certainly encourages you to keep looking closer.
Keep looking, keep questioning, and keep diving deep into the wonders of life.